Our body's tissues are aggregates of specialized cells that perform specific functions. The function of a tissue is directly related to the shape of the cells that make up the tissue. To learn about various cell shapes and functions, click on one of the buttons to the left. Erythrocytes, or red blood cells, are flattened biconcave discs that are about 7 microns in diameter and 2.2 microns thick. The unique shape of an erythrocyte provides an increased surface area through which gas can diffuse. Leukocytes, or white blood cells, are generally spherical to amorphic. Some leukocytes have the ability to change their shape in order to engulf foreign particles. Neurons have 3 principal regions: a cell body, dendrites, and an axon. Dendrites are thin processes that extend from the cell body. Dendrites act as a receptive area for stimuli and transmit electrical impulses to the cell body. The axon is a longer process that conducts impulses away from the cell body. Axons may vary in length from only a millimeter to up to a meter or more. Osteocytes are mature bone cells that secrete bone tissue. Osteocytes are located within spaces called lacunae. Canals called canaliculi connect the lacunae to one another. These canals house cytoplasmic projections from the osteocyte cell bodies. The canaliculi and cytoplasmic projections allow for the movement of metabolites and fluids between adjacent osteocytes. Smooth muscle cells are spindle-shaped fibers with a single nucleus. They are located in the walls of internal organs and are responsible for involuntary movement. Simple columnar epithelial cells are located throughout most of the GI tract. Many of the columnar epithelial cells found in the ileum have finger-like projections called microvilli. The microvilli can increase the absorptive surface of a cell up to 30 times its surface area. It is estimated that an adult human consists of about 75 trillion cells. These cells have much in common, yet cells from different tissues vary in a number of ways. For example, cells vary considerably in size. Cells and other microscopic structures are measured in units of angstroms, nanometers, and microns. The human hair, which is about 200 microns in diameter, is relatively large compared to the body's cells. The following illustration shows the relative size of various cells when compared to a human hair. Magnetic resonance imaging, MRI, is considered the best non-invasive technique for imaging soft tissues. An MRI uses a powerful magnet to generate clear, detailed cross-sectional images of the human body. MRIs can detect tumors too small to be seen with an X-ray or CAT scan. MRIs are also used to diagnose kidney, heart, and lung disease, as well as assess soft tissue damage to joints. Computerized axial tomography, CAT, is an X-ray technique used to create a cross-sectional image of the body. CAT scanners use a computer to assemble a detailed image from a number of individual X-ray beams. CAT scans are used for diagnosing a number of diseases, including brain tumors, aneurysms, and major artery disease. X-rays, discovered in 1895 by Dr. Wilhelm Conrad Röntgen, have become one of the most important diagnostic tools in modern medicine. A radiograph is produced by placing the body in front of a sheet of X-ray film. The body is then exposed to radiation from an X-ray tube. Dense structures such as bone impede the passage of X-rays, producing white unexposed areas on the X-ray film. Soft tissue, being non-resistant to the passage of X-rays, will allow the film to be exposed and blackened by the rays. Positron emission transaxial tomography, PET, is a technique used to determine the metabolic activity of body organs. PET scans provide a biochemical and metabolic picture showing the different rates at which chemicals are metabolized. These images are generated by introducing a radioactive isotope of a natural body substance such as glucose. The isotope is injected into the body, and its patterns of distribution are recorded by a computer. The computer then produces an image of the organ being scanned. Since normal and abnormal tissues absorb radiation differently, the images allow doctors to detect dead or cancerous tissues. Ultrasounds are valuable for visualizing internal organs and monitoring the size and shape of a developing fetus. Ultrasounds produce ultrasonic pressure waves that are directed into the body by a handheld device called a transducer. The ultrasonic waves are monitored as they reflect off anatomical structures with different acoustic properties. A sagittal plane divides the body into left and right portions. A coronal or frontal plane divides the body into anterior and posterior portions. A transverse or cross-sectional plane divides the body into superior and inferior portions. Medial refers to a body part being closer than another part to an imaginary midline dividing the body into equal right and left halves. The eyes are medial to the ears. Lateral refers to a body part being closer to the side of the body than another part with respect to an imaginary midline dividing the body into equal right and left halves. The ears are lateral to the eyes. Proximal refers to a body part that is closer to the point of attachment or closer to the trunk of the body than another part. The knee is proximal to the foot. Distal refers to a body part that is farther from the point of attachment or farther from the trunk of the body than another part. The foot is distal to the knee. Anterior refers to a body part that is more toward the front of the body than another part. The eyes are anterior to the brain. Posterior refers to a body part that is more toward the back of the body than another part. The brain is posterior to the eyes. Superior refers to a body part that is above another part. The trunk is superior to the legs. Inferior refers to a body part that is below another part. The feet are inferior to the legs. Superficial refers to a body part that is closer to the surface than another part. The skin is superficial to the skeleton. Deep refers to a body part that is more internal and farther from the surface than another part. The skeleton is deep to the skin. Ipsilateral refers to a body part that is on the same side as another part with respect to an imaginary midline dividing the body into equal right and left halves. The right arm and right leg are ipsilateral. Contralateral refers to a body part that is on the opposite side of another part with respect to an imaginary midline dividing the body into equal right and left halves. The right arm and left leg are contralateral. The life cycle of a cell can be divided into two distinct phases, interphase and M phase. A cell that is currently non-dividing is in interphase. Interphase is divided into three phases, G1 phase, S phase, and G2 phase. M phase is known as the cell division phase. M phase consists of mitosis and cytokinesis. Mitosis is further divided into four phases, prophase, metaphase, anaphase, and telophase. To explore more information on interphase and M phase, simply click on one of the buttons to the left. A cell that is currently non-dividing is in interphase. Interphase is divided into three phases, G1 phase, S phase, and G2 phase. Cells in G1 phase perform the physiological functions characteristic of the tissue in which they are found. Such physiological functions include the production of mRNA and proteins. Cells that are going to divide must first replicate their DNA during the S phase. During G2 phase, the chromatin condenses to form short, thick, rod-like structures called chromosomes. M phase is known as the cell division phase. M phase consists of mitosis and cytokinesis. Mitosis is further divided into four phases, prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes are seen as two chromatids joined by a centromere. Centrioles begin to form a mitotic spindle by first moving toward opposite poles of the cell. The nuclear membrane starts to disappear and the nucleolus is no longer visible. During metaphase, the chromosomes line up at the equator of the cell. The spindle fibers from each centriole attach to the centromeres of the chromosomes and the nuclear membrane disappears. During anaphase, the centromeres split and each sister chromatid moves to opposite ends of the cell along the mitotic spindle. During telophase, the chromosomes arrive at the poles and the mitotic spindle begins to disappear. New nuclear envelopes form and the condensed chromosomes expand. During cytokinesis, the cytoplasm of the cell divides, forming two new cells. In this histological image from the tongue, you can see an example of dense, irregular connective tissue. This histological image contains elastic cartilage, which can be found in the earlobe, auditory canal, and the larynx. This histological image contains fibrocartilage, which can be found in the symphysis pubis, the meniscus of the knee, and in the intervertebral disc. This histological image contains hyaline cartilage, which can be found on the articular surface of bones, in the nose, and in the walls of respiratory passages. This example of pseudostratified ciliated columnar epithelium was taken from the upper respiratory tract. Note the mucus secretions in this plastic embedded section. This is a histological image of a section of a villus in the duodenum. This is a histological image showing alveoli in section at high magnification. Although they are not distinct in this image, capillaries are found between adjacent alveolar walls. This image is from the recto-anal junction. Note the abrupt transition from stratified squamous epithelium to simple columnar. This histological image shows a section from the wall of the bladder.